Good evening. It's a pleasure to introduce Dr. Savitri Sharma, who is a medical microbiologist with very special interest in ocular microbiology. She is the director of laboratory services at LV Prasad Eye Institute, the entire network, and the president of Ocular Microbiology and Immunology Group US, which basically controls all the activities in ocular microbiology and immunology, including infectious keratitis. In fact, that is her area of special interest. She has a lot of credits. In fact, she was the past editor-in-chief of Indian Journal of Medical Microbiology, which is the flagship journal, and also the past president of Indian Association of Medical Microbiologists. And she has also authored a book on ocular microbiology. Without knowing herself, she probably has influenced a lot of people. In fact, as a resident, when I heard her first lecture at a ISA meeting in Delhi, maybe she remembers several decades ago, and some of my juniors and seniors, in fact, 5 of us got influenced, and all 5 came to LVPI, 2 as fellows and 3 as faculty. So that's how nice her lecture was, along with Dr. Rao, who was speaking on the clinical aspects. She spoke on microbiological aspects. And that actually made us aware that LVPI is doing such fantastic work back then. So over to you, ma'am. Thank you so much for making it to iFocus.
Thank you so much, Santosh. It's a real pleasure. And I thank CSF and this iFocus online. Actually, I got the mail and I didn't know this was related to CFS. So that's why I said I didn't get the link for the Zoom. But I'm very glad to be able to speak to all of you. So I would share my screen. All right, so I think everybody knows LV Prasad Eye Institute. I bring greetings to all of you. We are in 4 different cities in the country, and we have more than 120 vision centers with 12 secondary centers. So we try to do microbiology for the diagnosis of corneal infections as well as other eye infections at not only in the Hyderabad center, but in other centers also. So since I'm going to talk to postgraduate students, I would like to stop in between and ask questions. I also invite you to stop me if you like. Will they be able to, Santosh? Stop me and ask questions.
Yes,
and ask clarifications.
There are panelists, postgraduate students, they can ask you questions and you can ask them as well.
So let's make it very informal and interactive. So as clinicians, we all know that for any disease, a diagnosis begins in the clinic. And you first see the patient and observe the clinical features and make some mental impression about the disease by observations. And then you have some differentials which you would like to either prove or disprove, and then take the help of the laboratory. The laboratory in turn actually bases its initial observation again on what we say direct observation of the sample, direct examination of the sample. And in microbiology, since we are dealing with microorganisms, it has to be under the microscope. So then you put the clinical features and the direct observation together and again get some information. But this may not be always very sensitive. And then therefore we go for methods which will allow the organisms to grow and become more in number. And that's called isolation process, culture isolation. And then we try to characterize them and add value to whatever we have been trying to look for. Currently, everybody knows that molecular diagnosis has come into vogue. It's mainly because of the learnings of the scientists about the DNA and RNA of the organisms, along with the DNA and RNA of other human and other living beings in the world. The tests are being now directed towards trying to identify and learn about the organisms and also place that into the diagnostic methods. So obviously the tests would go by the etiological agents that one has in the mind, and corneal infections can be caused by any of these groups of organisms. It's not confined just to bacteria or fungi, viruses, parasites, and I hope I'll be able to take you through. I cannot go into very much details of all, but at least some things I'll be able to give you. Before I go further into the test, it might be interesting for you to see how the direct microscopy, which I mentioned that based on clinical features, you first do the direct microscopy and give the results as early as possible. So it's rapid and it's very rewarding if it is positive. So here is a patient, 50-year-old male, history of fall of unknown foreign body in the eye, treated with topical steroids before coming to us, and diagnosed as fungal keratitis elsewhere with certain procedures already done. And on examination, the vision is very poor. Cornea is perforated and shallow AC and boggy conjunctiva. Now you see that we took a scraping of the cornea from the cornea, and we find here the fungal filaments. It's very clear, it's very simple to make this diagnosis. Just you have seen under the microscope a very tiny, very small piece of corneal ulcer. And then I was actually, why did I include this case here? When I looked at, we have two smears to make, which I'll be telling you later. When I looked at gram stain, I am seeing only gram positive cocci, and see the number. It is very huge number. So I got a shock, and I thought, here there is so much of fungus, and this is a scraping, same patient scraping, gram stain should be showing the same fungus, but I am seeing gram positive cocci. And then I got worried that is it there is a mixing of sample, mixing of patient, or something has happened. But when I further screen the gram stain, I find that the same gram stain has gram positive cocci, plus there are fungal filaments here. So now I'm convinced that yes, fungus is there, not that there is no fungus in this patient. So this is a case of a very severe, very heavily infected fungus as well as gram positive cocci, and that is staphylococcus. So immediately our diagnosis is on the smear examination itself, which is within half an hour or 1 hour you can do. It is a mixed infection of bacteria and fungus. So this is what I mean by how quickly you can make a diagnosis by diligent direct microscopy of the corneal scraping. This direct microscopy observation actually has given us many other things. We have made, now I've been working in LV Prasad for 30 years. And prior to that, I was in LV Prasad Eye Institute for in Arvind Eye Hospital for 5 years. So during these times, we have been able to see many new things. Apart from acanthamoeba, which I had found the first case reported from Arvind Eye Hospital, I found microsporidia in the LV Prasad Eye Institute in the corneal scrapings. And you can see very tiny raised epithelial lesions which the patient comes with, and you take a scraping and you can demonstrate the presence of the microsporidia spores in the smear. You can also show on the gram stain, and this is calcofluoride. So what I'm trying to say is, by direct observation, you can find new organisms. And after we learned this, in 2003, I reported these cases. And after we learned, I have had 2 PhDs working on microsporidia. And if you go to either Google or to PubMed, majority of the articles on microsporidia are from our institute. It's just because we paid a lot of attention to this particular organism, and this is something which does not grow in culture. It is only seen by direct observation. Similarly, recent times, I have been very fascinated with keratitis called Pythium keratitis. It's caused by an organism, Pythium insidiosum. I'll be telling you more about this later. But this is a new entity. Again, the direct microscopy is very important. Though it looks like fungus, but they are actually not fungus. They are Pythium, and the difference I'll tell you later. And the treatment is very different. The fungal keratitis we normally treat with natamycin, and here you don't give antifungal, you have to give antibacterial antibiotics like linezolid and azithromycin. So I'm trying to emphasize that any microbiology laboratory should be spending considerable time and considerable efforts in looking at the samples directly as soon as they come into the laboratory and not just wait for the culture. But however, this technique may have limitations, and that can be seen here in this case, where we had a 39-year-old man who had redness, watering, and pain, and was diagnosed as or thought to be acanthamoeba endotheliitis somewhere else. But when he came to our institute, the clinician thought it was actually HSV endotheliitis, herpes simplex virus endotheliitis. And the clinician started the patient on steroid and acivir treatment for HSV. And then what happens? They take the scraping and we don't find any organisms. They again take scraping. We don't find any organisms on 2nd July also. But herpes simplex virus immunofluorescence assay was also negative. So there was no HSV in that. And when they repeated the third scraping, you can see here, sorry, I'll go back. We are seeing here acanthamoeba cis. So this patient was actually acanthamoeba, which in the earlier place it was thought of, but we could not prove it initially with 2 or 3 scrapings that we did. And we had to do the fourth or third one to find the acanthamoeba cis. So these are some of the limitations of direct microscopy. Now, to do microbiological investigations, we need very minimal things in the laboratory. You don't need a very big laboratory with the very fancy gadgets. All the items are listed here. To start with, wherever I went, when I went to Arvind Eye Hospital or came to LV Prasad, went to another LV Prasad in Bhubaneswar, then to GMRV in Vizag, wherever I went, these are the things initially you just collect for starting the lab. And basically we need a microscope. The normal microscope costs around 40,000-50,000. It used to be just 20 hydroxide and glycerol and gram stain, which in earlier days we used to make the reagents needed for gram stain, but now you can get ready-made from high media. You don't even have to make. So very simple things, all available very easily, can be put together to start a microbiology laboratory in an eye hospital or eye setup. Now let's see the microscope. This is a picture of this ordinary microscope. You can see that there are 4 objective lenses here, and they have different colors. These are universal colors. All over the world, every company follows this, and the colors actually indicate the magnification of that particular objective lens. The red one is the smallest one, which is a scanner objective lens, and it is usually 4x. Some people can have 2x also, but this is 4x. Red one is 4x. And then you have yellow one, which is 10x. And you have blue one, which is 40x. And then the last one, which is the biggest one, is the 100x. It is universal. Now, here is a table to just indicate, to get an idea what kind of magnification in terms of the microorganisms we can get. If you are using 4x, the eyepiece usually, which is hidden here, will have 10x. So 10 into 4, it gives you a magnification of actually 40 times or 40x. And any object under that objective lens, if it is 1 millimeter, in your imagination, if it is just 1 millimeter, its actual size is 25 microns. So if you just remember this, you can come down and see that anything which is looking like 1 millimeter and the objective you are using is 100, magnification is 1000. So the size of that object is actually only 1 micron. And we relate this to Staphylococcus. Staphylococcus is a bacteria. And the width of the Staphylococcus, the diameter of the Staphylococcus is a round cocci. It is just 1 micron. It is just like a one dot. Whereas in comparison, Candida is 5 micron in size. Fungal filaments are definitely more than 2 micron inside the width. The length can be very, very long. It can be anything, actually. We cannot measure that. But what we measure is the width. And RBC in our, from MBBS days, you know how big the RBC looks under the microscope. And it is actually 7 micron. So some perspective of the size of the organism is required to know what we should look at under which objective lens. This is just to show you the gram stain. There's no point going into details. Once you buy this or you come to the lab, go to any lab, we'll have these reagents and they will teach you how to do gram stain. Similarly, in gram stain, you actually see gram-positive organisms which are blue in color and gram-negative are pink in color. The other stains like Ziehl Neelsen stain also you are aware of, which is done for acid-fast organisms, and one can use two types of acid, 1% H2SO4 or 20% H2SO4, which will stain different organisms. If you use 20% and a slide is showing bacilli, which are acid-fast, pink in color, it suggests the Mycobacteria. Again, this also is very, very quickly, just in direct microscopy, you can give the diagnosis of Mycobacteria. And the other one is 1% acid is used for the Nocardia, which is a very thin filament, but it is branching. They are bacteria, but they are filamentous in nature. The beauty of diagnosing Nocardia and Mycobacteria in the direct corneal ulcers scrapings is that the treatment is very different. Immediately, you can give a specific treatment that is 2.5% amikacin eye drops, which are both effective for Nocardia as well as for Mycobacteria. So these stains will help you make the diagnosis very quickly. Until now, we talked about different aspects of setting up a microbiology lab and doing the direct microscopy and the benefits of that. So I would like the students or anyone to just say, was everything clear till now, and do you have any questions?
Ma'am, we have 2 questions on the chat box. Can I take them?
Yes.
Okay. The first question is, in a patient with keratitis already on a variety of antimicrobials, when do we send corneal scraping for microbiology? Is it immediately or 8 hours after the stopping the drug or 24 hours after stopping the drug?
No, you first do it immediately. And you evaluate the patient clinically, you send the first scraping immediately, because it is possible that despite treatment, we may be able to get the organism. And don't wait for. But only if we don't get anything and clinically the patient is worsening, then you can consider stopping the drug for at least 1 day. 8 hours stopping is not going to help, but at least for 1 or 2 days. Sometimes they wait for even 2 days and then they repeat it. But immediate one is very important. We have to do it immediately.
Okay, ma'am.
Okay.
The next question is, if a KOH mount is negative, what are the chances of getting a positive culture?
That will depend on which organism we are dealing with. So now I'm going to show you this slide, which is open now, is about the KOH and chalcofluoride. So you expect to see in this smear only fungus and acanthamoeba, microsporidia, the bigger organisms. You cannot see bacteria because that doesn't get stained with fluorescence, and we are looking only at up to 40x magnification, 400x total. So you will not be seeing the bacteria. So still culture can be positive, even if KOH is negative.
Yes. Any other hot seat participants? You can ask your questions if you have any. I think we will continue, ma'am.
Okay, so now this slide shows you we have stopped using only KOH, potassium hydroxide, because we have a fluorescence microscope like this. Fluorescence microscope has a ultraviolet source of light, which will actually create fluorescence of a fluorescent dye. You have to use a special dye, and that dye here is chalcofluor white. Chalcofluor white has a property of binding to the chitin of the acanthamoeba cell wall, the cysts especially, not trophozoites, and to the fungal cell wall. So because the chalcofluor white binds to those areas, under ultraviolet light, those areas will fluoresce. And that's how you are able to see very beautifully the presence of fungus here and acanthamoeba cysts here. We have also tried to find out some special stains, and there is a publication by Ruchi Mittal from our Bhubaneswar Center. When we started talking about pythium, see in potassium hydroxide with chalcofluoride, you can see very beautifully aseptate fungus. Can you notice that there is no septate at all in this fungus-like structure? And now we have learned, after seeing more than 100 and I think more than 160 cases of pythium, we have realized that we can diagnose pythium just on the direct microscopy by looking at aseptate fungal-like filaments. And we have always been successful in correlating that with the culture. Ruchi Mittal found another method. This organism is not fungus. It does not have ergosterol in its cell wall, but it has cellulose. And the cellulose will stain blue in color if iodine is added. So she has published it that you can use iodine for confirming that this is the pythium filaments. And here are some of the other pictures that she has published. You can read this publication where very beautifully you can see the blue filaments. Everywhere it is blue, whereas this is a normal fungus, which is not blue. It is yellow in color. So this is another stain for direct microscopy, which can give you the diagnosis of pythium. Now, there are papers, as you can see here, especially Western papers, which say that most ulcers may just respond to fluoroquinolone antibiotics or broad spectrum antibiotics. So why should we do smears and cultures? So in Western world, especially in the US, they are not very fond of doing or supporting microbiology or requiring microbiology for the management of their ulcers, not so much. But can we do the same in India? And now look at this prevalence of bacterial and fungal keratitis in different countries. You can see that the bacterial, which is the blue bars, are much higher in countries like UK and the US. But in US also, in places like Florida, which are warmer areas of the US, and then everything turns in the warmer countries. And in India, it is almost equal that fungus and bacteria are present. So if our patients are having either bacterial or fungal almost in equal proportion, there is no way we can escape doing the microbiological investigations and determining the cause of the disease for specific treatment. To rule out fungal etiology and treat specifically, we believe that even in secondary centers, we have many secondary centers, more than 12 in different parts of Andhra Pradesh, Odisha, and in Telangana. So we have placed in our secondary centers microscope as well as KOH and gram stain. We cannot have chalcofluor white there because the microscope is not a fluorescence microscope. So with these minimal things kept in these secondary centers and teaching the ophthalmologist how to do the direct microscopy, we are able to diagnose at least acanthamoeba and fungal differentiate fungus from the other types of ulcers. And here is a picture of different organisms in KOH, that is potassium hydroxide. So you can see fungal filaments very clearly. You can see Acanthamoeba cis. You can also see microsporidia. See, you have to reduce the light a bit and you can see very clearly the microsporidia. In Bhubaneswar, when I went first, we did not have fluorescent microscope, and most of the time I was diagnosing microsporidia only just in KOH. Nocardia also, with luck, you will be able to recognize. However, everything looks much better in gram stain because of the color, the bacteria, gram negative, gram positive, and here is a microsporidia, Acanthamoeba. Everything looks good in gram stain. One of our papers in British Journal of Ophthalmology, we have actually told about how you can use in secondary centers just the microscope, KOH and gram stain, and be able to diagnose many of your cases. You can see here that 46% of the total were actually fungus in that particular center. And this is now published. You can have a look at that paper. Other stains, sometimes they are used. I already mentioned about the acid pass stain. I'm very fond of Gomori methylamine silver stain. You all might have heard that in pathology, the fungus staining is actually Gomori methylamine silver. So what we do, gram stain, when we are looking at it and we think that fungus may be there, but it's not very clear, we give that slide to the pathology department and they do the Gomori methylamine silver, and then fungus becomes very clear with the green background. Black fungus looks very, very clear even in corneal scraping. So that's a very good stain. And currently we are actually trying to evaluate trypan blue, adding that to the KOH. Because KOH, as you saw the slides, there is no color. It's a colorless, it's not a stain actually, it's a colorless thing. So you will only find just a very light grayish organism. So if you add something blue in that, it will definitely become slightly better. And this is again to show you the nocardia, which I had already shown you earlier, and treatment I had mentioned, and these are the mycobacteria. This is our publication where we have compared these stains and we have shown that they are very good for fungi and Acanthamoeba, not so great for bacteria. Now here is a video, I hope it plays, how we take the scrapings in the clinic. So you do it on the slit lamp using a 15 number blade, and you take it multiple times, these scrapings, and place them on slides to make minimum 2 slides, and many culture media that I mentioned here. The blood agar you can take, and even chocolate agar you can take, and brain heart infusion, and SDA, that is Sabros dextrose agar and potato dextrose agar for the fungus. If you get tissues, like sometimes we get a biopsy sample, or even the corneal button comes to us, and many times we get bandage contact lens or something. All these items which are large, we cut into pieces and then place them onto different media for culture. Whatever grows on the medium has to be looked at carefully to determine whether they are really significant. And these are the criteria which I have actually written in many of my chapters and published in books also, which are very useful to me to determine whether the culture that has grown is significant or not. So for example, if there is consistency with the smear, you have seen something in the smear and it grows in culture, it is definitely significant. Growth in more than one medium, or very confluent or heavy growth, as you see in these plates, the fungus is growing so heavy, the bacteria is growing so heavily. So it is definitely significant, even if it is on just one solid media, but it should be in the area where you have placed the corneal scraping and not somewhere else. If something grows somewhere else here, where there is no sample, then that's a contaminant. You don't take that as the organism causing the infection. And in case of corneal scraping, at least it is possible to repeat sometimes if there is a doubtful growth. Clinically, you thought it is fungus, but we have grown only in one medium and we are not sure whether it's a contaminant. So we would like you to repeat it once, and so that we can see it also in the smear. Are there any questions until this point?
Yes, ma'am. We have two more questions.
All right.
In case of corneal ulcer, should Zn staining be done routinely for all these corneal scrapings?
No. We don't do, I actually, at one point, this question had come to me also, that if I do for all cases, I might catch more of mycobacterial infections. But no, it is actually counterproductive because it is the incidence, the prevalence of mycobacterial keratitis is very low. And to get one or two, if you start doing all cases, staining, it is very tedious and it is unnecessary. So what we do is, we clinically, the clinician decides whether it's an indolent ulcer. You have to read about the clinical features. As I said, the clinical feature comes first. So you look at that, and if the ulcer has been indolent for a very long time, waxing and waning course and not responding to whatever has been given, and then clinician wants to rule it out, whether there is mycobacteria. And that's when they ask us, or the other one, the clue, actually I went very quickly. In the gram stain, if you see unstained bacilli, we have published this also. You see the bacilli doesn't take the stain properly, gram stain. And shall I go back to that slide? Actually, I very quickly, here. You look at this gram stain. You have bacilli here, which are unstained. There is no color in them. And this here, there is looking slightly gram positive, but most of them are unstained. This is what gives me clue that there might be a mycobacteria here, because it doesn't take gram stain properly. There is mycolic acid in the cell wall and it doesn't get stained. So with this clue, which we have published actually in this publication, we have mentioned about this. And then the same slide, you subject to 20% H2SO4 Ziehl Neelsen stain, and you will find the acid-fast bacilli. So we wait for either clinical clue or microbiological clue to do the acid-fast staining. Now, one more thing I have to say. Suppose you have not got any of those clues and you have missed it in the gram stain, you could not. It grows on blood agar and chocolate agar, mycobacteria. So next day or after 2-3 days, you will find the growth. So we will not miss mycobacteria. By the method that we are following for culture, a combination of direct microscopy and culture will definitely give you the diagnosis. So we will not miss mycobacteria.
Okay, ma'am. Another question is, what is the specific indication of choosing PDA over SDA?
All right. You can manage with one, but I like to put two if scraping allows the number of scrapings that you can take, because PDA allows the growth of fungal spores much better than SDA. And so instead of having to go to PDA later, if you don't find spores on SDA, you may want to subculture on PDA. Instead of that, I prefer that initially itself you take one PDA and one SDA.
Okay, there is one little long question. At microscopic level, how differently does bacteria, virus, and fungi affect the cornea, and how differently corneal immune system react to them, which lead to different signs and symptoms?
This question is not related to this. I can't go into those details right now. We have time until what time? Till 9 o'clock. Yes, already
Yes,
half an hour is over.
Ma'am, I had one more question.
Hmm.
Ma'am, for GMS staining, which slide can we send? Do we need
Same
to take a
slide,
fresh
gram stain.
slide?
Gram stain.
Gram. Okay.
Gram stain, same stain. We don't have to go to the patient again to take another scraping.
Okay.
Gram stain. And same thing for acid fast stain also. The same gram stain, you can decolorize and do the acid fast stain. That's what I showed you. Those two slides are actually, it is the same slide. So that makes it very, very nice actually. You don't have to go to the patient. Patient might have, you don't have to give pain to the patient again and bother it takes time also. Again, going back to the clinic for that. Yeah.
There
So
is one more question.
now I'll move to, now till now, whatever I said, actually it is in the hands of the ophthalmologist. In a clinic, in your own clinic, you can do this. But now what I'm going to tell you is actually the job of the microbiologist. So if as a private practitioner, if you are going to go further into your investigation, you will need to actually tie up with a microbiologist to do the rest, that is to identify the organisms. It's not very easy for you to learn that and to have all the paraphernalia that is required for identification of the organisms. So how do we identify bacteria and fungi? You need to take many things into account, like the colony character, the rate of growth, what is the microscopic features, biochemical reactions. And now, very happily, we are actually having this gadget, which is called VITEK-2 compact machine. I'm very happy that all over India, many laboratories, microbiology laboratories have acquired this. This machine makes it very accurate identification of bacteria and as well as yeast, but not fungus. Fungus requires that we still look at the colony characters, the color of the colony. That's why it takes time for the color to come. The fungus may grow within 2 to 3 days. Like this one you see on a chocolate agar, this growth is just 1 or 2 days old. But for this color to come, this green color to come, it may take 4, 5 days or even 10 days. So that's why the species of fungus takes time to determine. And once you have a combination of the color, and then you have to make a judgment about whether it is fast growing and what is the color, and then you make a microscopic preparation from that, and then only you can say which fungus it is. And it is based on just physical characteristics of the fungus on the culture medium and under the microscope. Earlier days, we used to do a lot of biochemical tests for bacteria. We don't really need to do, but I somehow insist in the laboratory that though you have machines like VITEK, don't do everything with the machine. Then you will forget how the biochemical tests, the very simple ones, can actually identify the organisms. And also you will be not in a wrong track. If the machine detects something and gives a very exotic identification, at least you should have some basis for believing that. So my laboratory continues to do some of the biochemical tests, like mentioned here. These are for gram negative, citrate, utilization, urease, indole, etc. And this is the way the report of the VITEK comes. The VITEK will give you the probability. Like for example, this says Staphylococcus epidermidis is the identification. 99% probability, and it also says the confidence level, whether it is excellent identification or good identification. So what basically we have to do is, there is a card. You can see the card has many small cupules, and the culture which grows, culture has to grow, pure growth has to be obtained, and a suspension of that is put into each of these wells, and that is done automatically inside the machine. And then each of these wells actually contain the biochemical reagents and develop some color which the machine detects. And then against a profile of thousands of organisms profile, it will try to match the biochemical reactions and give you the diagnosis. So it is actually based on the biochemical reactions. Now, I mentioned about the utilization of molecular information for identification of bacteria and fungi also. So that is actually based on DNA sequencing. Now, every organism, including bacteria, fungi, have DNA and RNA. So we go for DNA. With our DNA can be isolated from these cultures as well as from the patient sample. Patient sample also you can isolate the DNA. There are procedures, kits available now. And you just take out the DNA and do PCR. I'm going to tell you about that. And then you can give it for sequencing. The sequencing means what? The DNA is double-stranded, and each of the strand is made up of bases which are nothing but A, T, C, G. Adenine, guanine, cytosine, thymine. So just these 4 letters, the sequence of these 4 letters as shown here and also shown in a graphical manner will tell you what is the sequence of the DNA. Now the DNA are actually, it's a very long stretch of DNA. You only choose one particular gene of the organism and try to amplify a small portion of that gene. You cannot amplify the whole organism, whole DNA. And that small portion, once it is sequenced, we know this sequence, and this sequence it utilizes bioinformatics for saying from which organism it has come, which organism makes this sequence. So let's see the next one. I'll come back to that sequencing again. But before that, let me tell you yet another method. And this method is based on detection of proteins in the organisms, not DNA. I hope you remember that they are very, very different. The protein of the organisms will also vary from one to the other. And this is a machine called MALDI-TOF. Some of in Hyderabad, I think 2 or 3 labs are having this. Each one is more than 2 crores, this machine. So we don't have it, but you can give your culture to these labs, and they can, based on the protein profile, they can identify the organisms. So they are also very, very reliable. Let's see antibiotic susceptibility. What is this method? What's the name? Another name for this? Can someone say? I've written here Kirby-Bar disc diffusion.
Disc diffusion.
Yeah, I've already written here disc diffusion. Okay. So this is a disc diffusion method. Now these are the discs which contain antibiotic. They are commercially available. They may vary in amount. For example, ciprofloxacin disc will contain 30 microgram, whereas chloramphenicol disc will contain 5 microgram. These are all determined earlier by the companies. And we buy the commercial disc. You grow the organism on the plate. This is a Muller-Hinton agar. This may be a staphylococcus which is growing over it. Now what we do is you spread the organism, grow the organism in a liquid medium, spread it and place the disc simultaneously. You spread the organism and place the discs. Then incubate the plate. As the organism is growing, the antibiotic from the disc is diffusing into the agar, isn't it? And if the organism is sensitive to this antibiotic, it will be killed wherever, up to where the antibiotic has reached. So if the organism is very, very sensitive, I like to use the word susceptible. If it is very susceptible to this antibiotic, it will be prevented from growing at a very low concentration also. So bigger the zone of inhibition, this whole round inhibition zone is called zone of inhibition. Bigger it is, more susceptible the organism is. Now in this case, it is not susceptible to it. So, and there is a chart where the level of, or the millimeter of diameter of this zone of inhibition is correlated with susceptible, intermediate, or resistant. So that's how we determine, and this is the easiest way to do. The other sort of slightly difficult way is to determine minimum inhibitory concentration, where we actually take different concentrations of this. One row will contain one antibiotic, different concentrations, and you place the organism in those wells and look for growth. Now you can see that there is no growth here. This is susceptible, this is also susceptible, and there is growth here, so it is resistant. So this concentration, which just inhibits the growth of the organism, will be called minimum inhibitory concentration. So that is that can also be determined by another test called E-test, where we have strips like this, which are available, and you can see this is moxifloxacin. MX is for moxifloxacin. And the antibiotic is in different strengths. Here it is 0.002, 003, goes up to 32 microgram per ml. That is a strength. And this strip, when you have placed here on the blood agar, and there is one organism which is inoculated here, you can see that there is a zone of inhibition which is egg-like or a teardrop-like. It's not round, but it is touching. It is touching somewhere on the strip. And this area, let's say it might be here, and it is written here 1.5. So this indicates that the minimum inhibitory concentration is 1.5 microgram per ml. Whereas you see this one, they're totally resistant. These are some other antibiotics. I'm unable to read. Cefazolin and something else is here. This also is quite resistant, but these two are susceptible. The organism is susceptible to these two antibiotics. All right. Identification of yeast is also done in the same VITEK. This is the VITEK machine, and some biochemical tests also I like to do, which can be done. Now let's come to the parasite acanthamoeba. Acanthamoeba does not form colonies, and which is the medium it grows on? Non-nutrient agar. It's written here, non-nutrient agar, which is nothing but agar in water. Non-nutrient is not a special medium. It is just agar in water. And you make 2.5% of that, and a solid surface will be there. On that, you spread some E.coli. E.coli you can easily get from a stool culture, or standard strains are also available. And then a corneal scraping is placed in the center of the plate, and no colonies are formed. You have to see under the microscope. So when you see under low part, you can see that there are some tracks here. Acanthamoeba is in the form of either cyst or trophozoite. The trophozoite is what grows in the medium, and it is motile. So it will move from one place to the other by pseudopodia. And this movement on E. coli forms tracks. So you can see these tracks, but we don't even worry about looking for the tracks because it's very tricky to see under the microscope. What we see is just the growth. You can see so many of these cysts of acanthamoeba forming clumps on the medium. You can very easily recognize them. And under high power, you can see the trophozoites also. So it's not really necessary to look for the tracks. One can actually recognize acanthamoeba on NNA very easily under the microscope. Any questions so far? Because I'm changing gears.
Yes, ma'am, we have few questions. How does the microbacterial growth of blood or chocolate agar look like?
It looks like small, very tiny, white or grayish white, slightly raised tiny colonies. And we confirm that it is Mycobacteria again by making a stain. We do 20% acid fast stain, and then we do some biochemical reactions.
Okay. One more question is, after testing, how are culture media destroyed?
Okay, so they are infective. So we have procedures for that. We have actually red bags in NABH, National Accreditation Board requires disposal in a particular way. So we put them in red bags and they are autoclaved and then they are discarded. A separate disposal autoclave you have to keep.
Okay. Ma'am, what is the use of double staining and what is the method?
What is double staining? I don't know. Did I say double staining?
No.
I don't know double
One
staining.
of the student was asking.
Okay. So let's see at this. Apart from bacteria, fungus, I have not yet come to virus, but there are other things which you can see in the eye. And these are some of the parasites. So this parasite is actually intravitreal. So let's not look at this. But Demodex, you must have heard of, causes actually blepharitis, but can cause keratitis. And this is actually a mite. You can see the mite. These are eyelash. Actually, the examination of the eyelash, which is epilated, and just in I put it in glycerol so that it will remain wet for some time, and you can see them very easily. Very huge parasite. This is a little bigger one here. They can be easily seen, and also one can see ectoparasites like blepharitis, you can see the Thymus pubis. Many times one can find. I have a nice video. I thought I'll just show you, where in the eyelashes you can see the nits and the parasites. These are the nits, and you can see the louse also. Here, this is the big louse here. Okay. So if these are there, they can be associated with keratitis. All right, now we move on to viral infections. The viral infections are mostly diagnosed clinically. Of the cornea, HSV is the most common virus. Very rarely, if you make an attempt in corneal scraping, you can see multinucleated giant cells and lymphocytes, which may support the diagnosis of a viral infection. If it is a dendritic ulcer, it is clinically very, very clear. So normally the cornea specialists don't go for investigation if it is a very clear clinically HSV keratitis. But in case of doubt, one can take corneal scraping and subject it to the test that we are doing is this one, immunofluorescence assay. And this shows you the principle of the assay. So let's say the red here is the corneal scraping of containing epithelial cells and which has the virus inside that. And then we have an antibody commercially available, which is anti-HSV. It has to be anti-HSV to bind to the HSV virus in the epithelial cells. And if this binds, there is a washing step here. Suppose there is no antigen, then if you wash here, this will go away and the test will become or come negative. But if it binds, then there is another reagent called anti-gamma globulin, which is an antibody for this antigen. This is antibody for this, but this is antigen for this. And this one is labeled with a fluorescent dye. So that's why it is an indirect fluorescent assay. It is not directly. If the dye was here in this primary antibody, and this is a secondary antibody. So if primary antibody was labeled with a fluorescent dye, then it would have been a direct fluorescent antibody test. Otherwise, it is an indirect fluorescent antibody test. And what you need is a fluorescence microscope. And this is how the corneal scraping will be positive if it contains the HSV virus. Nowadays, we don't do the culture. Earlier, I used to do the isolation of the HSV virus, and that we would do in cell lines. So this is a cell line. We can use many types of cell lines, but this one is actually human corneal epithelial cell line. And once the sample is inoculated and the virus grows, you can see cytopathic effect. And take some of these cells, and again, by the immunofluorescence assay, you can show the presence of the virus inside this. But nowadays, this is a very tedious method. We don't do that. And what we go for viruses is actually the molecular methods. These are very sensitive and specific, and they are very, very good. These methods are good for viruses, especially because they are difficult to grow. It's time consuming to grow. The laboratory will be very, very expensive where you can grow the virus, and many places it is not available. So molecular methods are the ideal methods for diagnosis of viral infections, and PCR is the most common procedure that is done. PCR, which is polymerase chain reaction. And I'll ask you questions after showing you this video. Watch carefully. The DNA divides into two. These are the steps of PCR. So what happened? When DNA divided into two, the temperature was around 94. Now here there is a thermometer. So around 94 degrees. And you are seeing these four colored substances in the fluid. Now what are these four? Can someone say?
Ma'am, different bases.
Yes, very good. So these are different bases that you have to supply in the reaction tube. And what is this yellow one?
Primer.
No. Primer is, I'll show you the primer. You can see primer is nothing. This is a primer, this small one.
DNA ligase?
DNA polymerase.
Yeah, that's a DNA polymer.
DNA.
Very good. So everything you have to provide for the DNA to amplify. And DNA is extracted from the sample, patient sample. And you give that, and you give all these bases and all the reagents required, and then one strand will become 2, 2 to 4, exponential increase in the amount of DNA. So now this DNA, which has formed or amplified within the tube, we have to observe. How do you know that that DNA is there? So this is the method which is gel electrophoresis we do. And you have many lanes in this agarose gel, 1.5% or 1.8%. And you can see now, I'll just describe this gel to you, where you have the negative control. There is nothing here. In positive control, you have a band here. Now, DNA binds with ethidium bromide, and ethidium bromide under UV light will fluoresce. It will just be brightened. So we add ethidium bromide to the gel, and when DNA passes through that, DNA of same size will settle in one place, and then in the machine, which is called gel documentation machine, we put the gel and it will show you. And nowadays they are very, very sophisticated. You put the gel inside and the whole machine is connected to a computer monitor. The picture will be shown on the monitor actually. And then this is the picture you will get on the monitor. So you are finding there is a band here in the positive control. This is one patient sample, similar band is there. This is another patient sample, no band is there. And here there is a marker. These are different sizes of the bases. This is 100 base pair, and then 200, 300, 400, and 500. So you know what exactly is the base size of what you have got. Now you can see this is 100 and 200, and this is somewhere slightly above the 200. So the size of this DNA is between 200 and 300. So this is meant for measuring it. And once we get this, you need to purify this if you want to do sequencing. Otherwise, this is enough to give us the indication that if your positive control is that of herpes simplex virus, your sample is also containing herpes simplex virus because it is at the same place. And the primer we have used is always a very, very specific primer. The primer positions on the gene are known, which type of gene you have used, and what is the sequence in the primer. Everything, this is all theoretically one has to standardize and then do the PCR. And that's why theoretically you know that whatever is the product size will be 221 base pairs. Exactly to know that it is 221, here you are already knowing indirectly because it is between 200 and 300. But to know exactly it is 221, you will need to do the sequencing. There are other modifications of PCR, multiplex, real-time PCR, DNA. This I'll be talking to you about real-time PCR also. I'll tell you a little bit. When you do normal PCR, whether there are of a particular gene, whether amplification has happened and it is only 10 copies or 100 copies, it will not make much difference in the thickness of the or the density of the band that you saw, unless it is very, very drastically different. It will look almost the same. But when you do real-time PCR, what one does is to the DNA, which is amplifying, cyber green is a dye which is used. It binds to that DNA. And so as the DNA is amplifying, the machine will detect how much of DNA has been amplified. So it gives a measurement of amount of DNA. So everything else is same. But it will give you a copy number. It will give you how many copies. And there is now, I think with coronavirus disease, you are reading about RT-PCR, which is a real-time PCR of reverse transcriptase real-time PCR. So you are also reading about what is called CT, cycle threshold. And the moment the DNA starts amplifying, when it starts amplifying, it crosses the threshold. So the lower the threshold, more of DNA. It is inversely proportional and more of copies. So lesser CT means more DNA or more virus. Now currently there was a regulation from ICMR that the report of RT-PCR for coronavirus should also give the CT values. And the way you read the CT value is lesser it is, more of DNA copies are there. You can get exact copies of DNA also by having a corresponding standards. So you have to have a standard where known copies are there. You make a graph from that and then extrapolate the findings to that graph, and you can derive the number of copies. This is just to show you the paraphernalia required for doing all this. You have to have a laminar flow hood for DNA extraction, other machines required for the procedure of DNA extraction. And this is the PCR machines. 2 machines are here. Gel electrophoresis, which I mentioned to you. And this is a gel documentation system I mentioned. You put the gel inside this because there's a UV light inside that. We don't want UV light to be exposed to outside. So after putting the gel through this door, you close it and then switch on the UV light, and the whole picture will be on this monitor. And this is a machine we have of real-time PCR. These are all pictures from my lab. So a lot of investment goes into be able to do the molecular diagnostic tests. Any test you do, whether it is PCR or others, clinical correlation is the hallmark. Ultimately, it has to make sense for the management of the patient. DNA sequencing, how do? We have the sequencer. This is a sequencer machine in our institute, but nowadays there are many companies outside. You extract the DNA, you do the PCR, and then you can send the PCR product to them. They will do the sequencing, and by email, they can send you the results, which I already showed you in the other slide, which is nothing but the sequence of the ATCG. And ATCG put together can look something like this. Everywhere you see it is just ATCG, but this is the sequence report which comes to you. And you can just copy this, and the bioinformatics which I mentioned to you is a science by itself, which many of us are, at least I'm not very familiar with that, but I understand how it is done. You copy this and you actually put it in a gen bank, which is actually available online. You can put the sequence, and then there is an icon called blast. When you press on that, you find that it gives you a list of organisms to which the particular sequence matches. And these sequences are all there on the websites in public domain and freely available. We don't have to buy them. They are freely available. Now, how does this get built in the system? You need, do I have more time?
Yes, ma'am, please continue.
Okay. So how does this get built? How so many DNA sequences are available on the websites? So I must, it's very interesting to know that suppose I'm doing work, like for example, I'm working on one organism called Pythium, and I have sequenced one of the genes. The moment I sequence, it is mandatory for me to put the sequence on the website. I cannot publish my sequences without first putting it on the website. And the website will give an accession number. That number I must include in my publication. And when somebody reads that paper, they see that accession number, they can go to the website, put that accession number, the whole sequence which I have put on the website will be available to anybody. So it is very nice. The scientists have built such a website, and it is called GenBank in the US. It is called something else in Europe and Japanese. these three organizations are maintaining this website, and all over the world, every scientist is bound to submit the sequences of anything that they sequence, whether an animal or an elephant or a bacteria, whatever they sequence, they're supposed to put the sequence on this website, which is freely available to everyone. So once you put any, now this is the sequence which I got for the gene which I was studying, and I'm expecting that it is a gene of the herpes simplex virus. So I put this on the website and then say blast, which will look at all the genes sequences which are available on the website, and it will give you a list of what probably it is matching with. And you can see that the first one that shows up is human herpes virus 1 strain 17 complete genome is what it is matching with. And here there are other values. What is a query coverage? What is a e-value, total score, everything. So with all this, we know that my DNA, which I had amplified from the patient sample or whatever sample, is actually 100% likely to be human herpes virus 1 strain. So this is how things are done. This is actually very brief. There is much more in this, which we are not always there is no time for us to discuss about that, but this is, I'm very happy to show you that using this technique is what I could confirm that the Pythium, what I'm calling Pythium, is actually Pythium. So you take the sequences and you can see all these blue ones are the isolates which from our laboratory we have got, and the others are already there on the website. So there are software which will place your isolate, how close it is to the others, and it shows you that it is very, very close to so many other Pythium insidiosum which are there on the website. And that's how you confirm. And this is already published, what I'm showing you. This is called phylogenetic tree. It measures the closeness of your isolate to the ones which are already there in the website and tells you that it is how close to those which are already there. So mine are very, very close to the other Pythium insidiosum sequences which are there in the website. And now we know that the direct scraping of cornea will show Pythium like this. The colony looks like this. So actually for me, I first learned by doing DNA sequencing that I got a Pythium. And only later I learned that the smear looks like this and culture looks like this. And now stick features of the patient so that they can catch the diagnosis just by looking at the patient. And this is a histopathology picture of that. I use a method called zoospore. You can see the zoospores of the Pythium coming out of this particular thing. And I use this in the laboratory to confirm the Pythium which grows in blood agar very easily. The reason I'm doing this is it is 100% specific for Pythium insidiosum. And without doing sequencing, by this simple method, we are able to say that the isolate we have got is Pythium insidiosum. And this technique we have already published in the paper that we wrote in Cornea. It's come in Cornea. This is just, I think I have short the time already. This is PCR of Microsporidia, not really required because we see very nice spores in the direct microscopy, and that's enough for diagnosis. We don't really have to do this. Deep DNA sequencing, this is not the time or place for me to tell you about this. I just want you to know that people are now working on what is called next generation sequencing. So this is nothing but instead of this normal PCR, which deals with very short fragment of the gene, in next generation sequencing, one can actually make fragments of a very long gene and do the sequencing of the whole length of the gene very easily. And this technology is also available currently, and many people are now, many microbiologists are now trying to use this for diagnosis. You might have heard of DNA chips, which were actually made by the Exciton company in Bangalore. We had participated in trying to develop that along with Shankar Nethralaya and RP Center and CCMB. So this is the kind of the chip that was developed, and one could, this is also a DNA technology, a modification of, it requires PCR and then you have to do hybridizations. It's not possible to go into details. Another kind of test you may read somewhere is LAMP assay. And last slide, I think before the last, I would go for the limitations of the molecular methods. Not everything is not hunky-dory. Training of personnel is very, very important. Expensive initial setup, effective control of the parameters, quality control is very important. And commercial kits are considered better than in-house developed techniques. And finally, clinical validation. Clinicians have to participate in that to let us learn which tests are really useful for diagnosis of the clinical condition. And there is a demand for point of care tests similar to what you have for adenovirus. I think you must be already knowing. These are like pregnancy tests. This I already talked to you. And now even for coronavirus diagnosis, antigen detection test is actually a point of care test. You can just take the nasopharyngeal swab and put it in a buffer and put it in something like this, and you get a line. So there will be a control line, and you get another line, and then the test is positive. So the microbiologists are working towards these easily available point of care tests. And here is one which is also available for herpes simplex virus in Japan. So with this, let me just tell you, there are lots of advantages of going for molecular methods. It rules out, because it is very, very sensitive. If it is negative, it rules out the infective etiology. It's rapid and definite diagnosis of infective etiology. It can make reliable confirmation of unusual organisms, new organisms you can find by these methods. And it confirms the presence of organisms which are difficult to confirm by other means. So with this, I thank you. And here, this is to show that I really don't go for PCR for acanthamoeba, though you may see in the literature, because this is what I have published. The direct smear examination gives me as good a diagnosis as PCR. So this is a comparison between smear and PCR. And you can see their sensitivity, specificity, and predictive values are almost similar. So we don't really go for PCR for everything that we are doing. With this, I thank you very much. I'll take questions if you have any more and if you have the patience.
Yes, ma'am, I'll take the questions. Can we identify the coronavirus by sending the samples of conjunctival swabs and the secretions, the tear secretions, the PCR?
Yes, I think just yesterday I read. Before that, what I have read is many patients of corona disease are actually coming with conjunctivitis, and they are not able to show the virus in the conjunctival fluid or the conjunctival swab. But just yesterday, I read a paper has come where 3% of conjunctivitis, patients having conjunctivitis, having coronavirus, only 3% showed the presence of virus in the conjunctival swab by RT-PCR. So
Okay.
some percentage they are able to show.
Okay, ma'am. Another question is, do we always need to culture to do PCR?
No, not really. PCR can be done with a clinical sample directly. There are two ways. One is you do with the clinical sample, and the other one is when we want to identify an organism which has grown and we are unable to identify by biochemical tests and other things, we can do PCR with that also.
Okay.
But for clinical purposes, it is clinical sample.
Okay, ma'am. The next question is confocal microscopy versus corneal scraping. What do you recommend for effectively monitoring the antifungal treatment?
Definitely confocal. If you have that for monitoring treatment, it is a confocal.
Okay.
Because once the cornea starts healing, then the organism, to get organisms in the cornea scraping and culture, it will not grow because it's under the effect of the drug. And epithelium starts healing, you won't like to disturb that. So I think it is confocal.
Okay, ma'am. Ma'am, I have one more question. How frequently do you test for the susceptibility of ceftaroline in MRSA cases?
What?
Ceftaroline, 5th generation cephalosporin.
We don't.
Okay. And next question is, what is the role of antifungal susceptibility test and when do you recommend it?
Yeah, we do it sparingly. One is you do for research, for many of the research projects we have done. But routinely we don't do unless the clinician feels that patient is not responding and would like to consider another drug. The thing about fungus, antifungals is that we don't have a big, many antifungals available. We have only few. So there is no profile that needs to be studied, unlike the bacteria. So we do it only on request.
Okay, ma'am. The next question is, how often do you get patients with mixed infection and what is your take on the broad spectrum management for the same?
For mixed infection.
Yes, ma'am, mixed infections.
Broad spectrum management, how is it related to mixed infection? Mixed infection around 5% in corneal ulcers. Our analysis has given around 5%.
Okay. Any of the hot seat participants have any questions to ask?
I have one question. Ma'am, in case we are unable to send any inoculated culture media immediately to any microbiology lab, then how long and in what way can we store culture medias like chocolate agar, blood agar, or SDA-PDA once they are inoculated?
See, once they are inoculated, sometimes you take it in the evening and you are not able to get it to the lab. Just yesterday we were discussing, you can have a box in which you keep it and keep it at room temperature. Don't keep it in the fridge because it has to go to 37 degrees, which if you're not able to get at least room temperature, which is 25, 26. It should not go into the refrigerator and should be in a closed box so that insects and ants and something should not go inside that. It should be in protected area.
And how long can we keep it? Supposedly we are in some
Not more
remote
than
areas.
overnight. Just overnight. Next day it has to go to the lab. Yeah, because
Okay.
the medium will start drying and you can't keep the organism alive on that.
Okay.
Not
And
more
ma'am,
than overnight.
is it possible to subject swab samples to PCR? If we have taken some conjunctival swab or something, is it possible to
Yes,
subject it
why
to
not?
PCR?
If nasal swab, you can do PCR. Why can't you do with conjunctival swab? But immediately the swab has to go into a buffer,
Okay.
not a dry swab. You take the swab and it has to go into a buffer.
Buffer like phosphate sulfur.
Yeah, phosphate buffer saline.
Okay. Thank you, ma'am.
Okay, any more questions? I think we are done with the questions, ma'am. Thank you so much.
Thank you so
Thank
much.
you very
I
much,
hope
ma'am.
it was useful.
Very, very extensive, very useful lecture.
Thank you so much.
Thank you so
It
much.
was
ma'am.
a pleasure. It was my pleasure.
Thank you so much. Thank you so much. It's a privilege to have you on this, ma'am.
Thank you. Thank you.
Good night. So next Wednesday, we have Dr. Prafulla Maharana from RP Center talking on interstitial keratitis. So please be there. That will be 16th September. Thank you so much.
Thank you. I log out.
Yeah, ma'am. Thank you so much.